Good morning, everyone. My name is Dr. Urvish Patel, and I work in Department of Public Health and Neurology at Icahn School of Medicine at Mount Sinai, New York, US. And thank you so much for giving us platform to present our project in the conference of World Congress of Neurology. Today, the topic of my talk is genome sequencing, mutational analysis, and survival rate of glioblastoma multiforme. We do not have anything to disclose. We do not have any fundings, and I appreciate my all contributors who helped me to complete this project. Glioblastoma multiforme is a deadliest form of brain tumor with the incidence of 3.21 per every 100,000 population, with very little median survival rate of 19 months, and overall survival rate is 2.2% over 3 years. Mainstay of the treatment is always surgery followed by radiation and chemotherapy, but research in the treatment is always revolving around three important aspects. Number one is specific gene mutations within tumor that can improve the prognosis. Second, that we can identify any or predict any response rate to the therapy and assess the presence of any experimental therapeutic targets. When we talk about different alterations or therapeutic targets in glioblastoma, we have some research from the past previously published studies in which they have identified IDH mutation, which is good for potential therapeutic target, MGMT methylation status, EGFR mutation, TERT is a promoter mutation, is a good also diagnostic marker for glioblastoma. We are having 7p gain and loss of 10q, H3F3A, and few more, which is one of the good identified, I can say, molecular alteration in the glioblastoma, which we can either target for potential therapeutic target or diagnostic marker, or somehow it will be helpful for us for the better management of glioblastoma people. Now, the gene TTN on which our today's talk is focused on, that gene TTN, which code for the protein retin, that is mainly expressed in the skeletal and cardiac muscle, and that is involved in many of the cardiac and skeletal hereditary and other disorders. And also one or two studies are out in which the TTN gene mutation were associated with the chemotherapy response in non-small cell cancers, carcinoma of the lung, and it is also correlated with the higher tumor mutation burden and better response to therapy. But that is only limited to the small cell cancer of the lung. We do not have any evidence or previously published article that can correlate between alteration and TTN gene and any survival in glioblastoma multiforme. And there is very limited literature highlighting the whole genome sequencing and its overall survival rate. So today, the aim of our objective is to analyze the genomic profile and identify the epidemiological characteristics of GBM. And secondary aim of our study is to identify survival rates at 12 and 24 months with the common mutations. In methods, what we used was the C-Bio portal cancer genomics. This cancer genome atlas, pan cancer atlas, and TCGA Firehose legacy. So this is bioportal cancer genomic directory, which we use to study the genetic mutation associated with glioblastoma multiforme. So C-Bio portal for cancer genomic was originally developed at Memorial Sloan Kettering Cancer Center, and it is publicly available with the hosted by Center of Molecular Oncology at MSK. We identify epidemiological characteristics and genetic profile using C-Bio portal database. A query was generated to calculate 12 and 24 month survival rate with most common mutations. Log-rank and Kaplan-Meier estimators we used to analyze the survival function, and we created, we did survival analysis, and we created the survival plots for glioblastoma multiforme patients. Now, the patient who is having 2 or more overlapping mutations, in our study we had 715 such people, patients. We excluded all of them to identify isolated effect. What we identified in the in our patients, that total 2041 GPM patients were there, out of which 1032, 51% were male and 32.5% were female. Out of those, 37.6% were white ethnicity, 4.1 were black, and 0.8 were Asian. So majority of the patient were between the age group of 50 to 70 year. And I can say in a different age group, we find out the percentage of the distribution is 9.1% between 50 to 55, 10.7 between 55 to 60, 10.1 between 60 to 65, and 8.3 between 65 to 70. So this is the major distribution of this tumor between 50 to 70 year of age. 78.8% were deceased, so they died. And by the time we have our analysis in the cross-sectional way, and 18.4 were alive. 95.1% had primary cancer, and 3% had recurrent cancer. The common mutations, we identified many mutations in the CBio portal registry, but the common mutations which we identified associated with the reduced survival were TTN, that reduced 7.6% survival, MUC16, NF1, PIK3R1, IDH1, EFGR, which is known, PTEN is again known gene, and TP53. So we identified the genes which is associated with the lowest survival rate. And out of this gene evaluations or mutation evaluation, some of the genes are new, like as I mentioned, TTN and MUC16. So these are some new genes which we identified. Overall survival after initial diagnosis of following 12 months was 58.9%, and after 24 months it was 24.2% respectively. So survival was lowest among TTN. So in the TTN mutation, after 12 months, the survival percentage was within a year 30% only population or patients I can say were alive, and after 24 months, none patients were alive. In P10, 36.4% patient were alive after 12 months, and after 24 months, only 9% people or patient were alive. Similarly, in CDKN2A mutation, 42.3% were alive after 12 months, and 15.4% alive after 24 months, and so on. So majority of the gene which is associated with the lowest survival, which we mentioned in the earlier in this analysis, followed by that whatever was associated with the higher survival that were in the later phase, like for example MDM4 and TP53, that is having 45% survival, I can say, at 45 and 84% survival in TP53, and MDM4 71.4 and 0% survival. The p-value is drastically statistically significant, 3.2 e raised to -10. So this survival is statistically significant. Here we are having Kaplan-Meier curve with long-range test with significant p-value. At 12 months, we are having unaltered group described or mentioned in the blue line. Compared to that, we are having TTN group mentioned with the red line. At the 12 month, the survival reduced in TTN drastically lower compared to other gene mutations. Similarly, in 24 after 24 months of survival rate calculation using Kaplan-Meier curve, the TTN, if you can compare here, almost at the month of 15, there is no survival. Almost every patient died at the month of 15, and also we are having lower survival here, almost zero, with MDM4, which we can correlate here. MDM4 after 24 months, it's having zero survival. So now we are having the list of the gene from CDKN2A, MDM2, MDM4, all the way to TTN, where median survival we can identify. That is, the Kaplan-Meier is like after 24 or 12 months, how many patients were alive or how many patients were dead. Compared to that here, whomever were alive, what was the median month of survival? So you can see TTN gene is having 2.7 median month overall. So that is very important gene, I can say, to target if we would like to continue our research. So TTN and PTEN are having lowest 2%, 2 month and 9 month survival. CDK2, this one, CDKN2A, 10 month of survival. So these are the few genes which is having very low or very less month of survival. So what we identify, or why we did this study, we wanted to identify some important gene which is associated with the lower survival, so which we can target as a new biomarker or new therapeutic agent, I can say, or therapeutic target we can see. So in our study, we identified TTN gene as a novel biomarker for poor prognosis of GBM, and that the alteration in TTN gene was associated with the worst prognosis and the median survival of just 2.7 months. In addition to that, PTEN, MDM2, MDM4, all three genes were alteration in that gene were associated with poor prognosis, with median survival within 1 year, compared to median survival of unaltered group, which is 19.6 months. So it is drastically reduced. The take-home message from our study is that GBM with TTN, PTEN, and MDM4 mutation were associated with the worst prognosis. And this study could create insight and may provide the foundation for developing new future therapies in terms of therapeutic targets for these patients. And here are some of the reference we use for our study. And I would like to thank CBio Portal for cancer genomics to give us the data, World Congress of Neurology and World Congress of Neurology Conference to give us the platform, and our senior authors and other mentors from Stormont Well and Mayo Clinic to give us the guidance to complete the project. Thank you so much.